All right, so in this class I'm talking about catabolite repression and regulation by branched amino acids. I'll just look at the papers. So in papers, the first paper I'm going to talk about is talking about Bacillus subtilis, and it's going to talk about the acetate kinase gene and how it's regulated by COD-Y and CCPA. This is done in Tufts University at Boston. Next paper is a review of the networks in Bacillus subtilis. I just wanted to show you the graphs and the figures from this paper. I didn't want to go into much detail about it. I just thought this was kind of an interesting picture to look at. Next one, in mutans, they looked at the regulation of COD-Y and CCPA by aminotransferase ILVE, and last paper, COD-Y activates a small RNA, and this paper is out of Germany. So the first COD-Y,
COD-Y, it's no, it's called DNA update.
Oh,
This COD-Y.
okay.
In step mutants also it's
the name COD-Y comes from a small molecule coid. It's the, oh my god, I can't remember, coidine, something like that. Operon regulator. Yeah, it's not the copper.
no, it's not COD-Y.
So the first paper is by Robert Shivers. Let's take a look at that. What is COD-Y? COD-Y is described as a dipeptide permease, and it is found in a lot of gram-positive, especially in firmicutes, and it regulates 100 genes.
Permease operon repressor, not dipeptide permease, is it?
Operon repressor, sorry, yes.
So it represses this operon.
Yes.
Okay.
It represses genes. It's found to be both a repressor and an activator.
Am I confusing it with something with in strep mutants which is involved in DNA uptake? It's also called COD-Y, right?
DNA uptake by COD-Y? I don't think so.
I think it's stress, right?
It is stress. So it might be involved in stress somehow. Bernie, COD-Y, yeah.
Yeah, it's possible.
Oh, there's a COD-Y paper involved in pentaphosphate, guanine pentaphosphate. And COD-Y is sometimes activated by GTP, isoleucine, valine, like the isoleucine and valine, they're the branched amino acids. And in Bacillus, it's activated by GTP. They haven't figured out if GTP works in S mutans. So for the tests they've done, they couldn't show that. But they could show that isovaline and isoleucine, and valine both are active in it. In Bacillus, this protein's a dimer. It dimerizes. It shows the helix turn helix with a C-terminus wing shown here at the bottom. And in this protein structure, what you have is inside of the protein binding an isoleucine. Also, a motif is required for this DNA binding protein. Like most transcription factors, they require a inverted repeat or a palindrome to have this activity. CCPA is involved in global regulation, and it's the catabolite repressor. It binds locations on the genome called CRE sites, the catabolite repression elements, CRE. And what it does is it represses genes for different carbon sources. Especially in Bacillus, they were talking about the Krebs cycle. And I like this little image here. It shows if this is the CCP domain, and here's your helix turn helix, and this is DNA. This is a really nice kind of picture, so let's take a look at that at Protein Data Bank. Acetate kinase, amino transferase, this one. Yeah.
Excellent job. Your computer is not connected to internet.
Sure it is. I closed the lid and it forgot who it was connected. So here's the CCPA protein. I just put it into its two subunits. As you can kind of see there in purple at the bottom, that's the DNA bound to CCPA. And you can see how on the edges, so if this is the center is CCPA, on the outside edges, this protein interacts with another protein called histidine protein, or HPR. So CCPA and HPR are bound together here, and they're also associated with DNA. And in this case, you can even make out, I don't know if it's really in good shape on the bottom, but you can see where it's binding to the major groove and the minor groove. So the DNA binding region is binding the motif there. In this case, it's binding a catabolite repression element. So that DNA is actually one of these elements. There's also a protein called acetate kinase, and acetate kinase, well, it's part of the TCA cycle, and what happens is acetate's made from pyruvate by acetyl-CoA, by pyruvate dehydrogenase and acetate kinase. And the acetate kinase genes are positively regulated by CCPA. And like I was saying earlier, COD-Y targets are usually repressed by COD-Y, like ACKA is repressed by COD-Y, but some are also positively regulated. And this paper tries to figure out, does COD-Y act on the promoter, or does it control ACKA independently? Like, how do they work together? Do they work independently? So that's kind of the question. So here's a TCA cycle involving COD-Y and CCPA. So as I was saying, pyruvate forms lactate, and COD-Y activates lactose dehydrogenase, CCPA activates it. And on the other side, CCPA represses the formation of acetate and acetylene. So it's really sensing and determining what the cell can do with carbon. Okay, one of the experiments in this paper, what they were looking at was what are the effects of COD-Y and CCPA mutations on the acetate kinase expression. So if you take wild type, COD-Y and CCPA wild type up here, so they do a beta-galactosidase activity assay, and this is what it's like for wild type. Then they take a deletion.
Transcription or translation of ACPA?
Expression. So
Transcription.
Transcription. So then what they do is they take a deletion of CCPA, but wild type COD-Y. So that's the first one. And notice the activity is really down. So one gene deletion, the activity is down. Same thing with COD-Y deletion, it's really down. And if you delete both genes, it's even further down. They do a similar kind of experiment. Okay, if you add or don't have isoleucine valine in the mix, if you add isoleucine or valine, you get expression going up. But if you remove it, it goes down. And here's, I was saying COD-Y is named for a compound, and here's the name of the compound, decoyinine. That's kind
Which
of a,
strain are they adding these branching amino acids?
beg your pardon, which strain?
Yeah, wild type.
In letter B, yeah. So wild type ILV, it's going up, and if you take out the branched amino acids, it's down, and if you add decoyinine, it's going down.
So why is it not showing as we have it in A? What's the difference between
A
wild
is
type?
just the expression of these genes, and down here, it's the expression of wild type in the presence of these effectors. And what they do is they make an LAC-Z fusion with the acetate kinase gene, promoter. So by this they can say having expression upregulated by branched amino acids, it's also requiring COD-Y and CCPA.
One more question.
Yeah.
So in the B figure, they are adding extra of these branched chain amino acids, right?
Yeah.
So in figure A, they were not using any amino acid. Why is it not going to the level of, as you see the scale?
100, yeah.
Here it's 100 for wild
Actually,
type without
if this is 100, 124,
no, it's 300, right?
it's hard to make out on this graph, isn't it?
Okay. I don't know.
I know this is 100 here. 120, 140, 180, 200 maybe at the top.
Are they the same scale, both of them?
They're activity units.
Yeah. Same scale?
I think so.
So what I'm trying to say is wild type there, it's having this uniform upregulation, like they are having expression of this gene, right?
Right.
For the wild type, and it's just constant, it's plateau. Whereas here, without isoleucine, without branch amino acids, the activity is at 80, and with amino acids, it's going up, and gradually it's going to
Right.
that
So
level.
perhaps this mix has the branched
Yeah.
amino acids in it already because it starts off at this point,
Yeah.
80,
And there it's starting
and it's
from,
going up to where it started here.
Yeah. Okay. I don't know. Just saying.
Do you have a question? No? Okay.
She has a question. And then I'm
Next.
finishing.
Well, I didn't check the time. I don't want to take too much time. Next, so they're doing in vitro binding of COD-Y to the ACK promoter. They're doing EMSAs. So in the case where you're not adding an effector, you don't get any complex being formed. Well, maybe some sort of complex, but not much. Same thing with a little bit of GTP. There's a little bit of evidence of a complex, but it's kind of weak. Now if you take branched amino acids, you get a really nice complex being formed when you're adding this much amino acid, 10 millimolar, and maybe 150 nanomolar COD-Y. So there's a nice complex being formed with the promoter to COD-Y in the presence of the branched amino acids, and it seems to be enhanced with GTP at the lower levels. But they don't consider this to be relevant.
So they are just
kind of miss
taking
that.
proteins, right?
They're adding protein to the promoter.
It's in vitro.
Yeah.
It's in vitro, right?
And what they're doing is, this is a radio-labeled gel. So
What are they trying to show here?
that ACKA promoter binds to COD-Y.
But only in the presence of
Branched amino acids.
Branch amino acids and branch amino acids, not much by GTP. So are they saying that branch amino acids go and bind with any of these, and that's why they can bind together?
the branched amino acids are required to bind for COD-Y to form its active complex. And
On
then,
protein level.
yeah. So COD-Y is a protein,
Yeah.
and ACKA is the DNA, and the DNA is what they're resolving.
Yeah. And if you're adding amino acids, it's also on protein level.
Yes.
Okay.
Yeah. So you could do this at a protein Western type experiment as well. However, they don't have the antibodies probably. All of this was done with expression data. It wasn't done with any antibodies in this experiment.
DNA expression? What expression did you?
RNA expression, DNA, RNA. It's all at the DNA, RNA level that they're looking at.
They're doing M-cell.
Yeah.
So it's between protein and DNA.
Right. But they're resolving the DNA, not
Okay.
the protein.
Yeah.
Okay, next picture. All right, what they want to do is they want to show, does adding COD-Y to the promoter bind someplace along its DNA site? So they have this sequence, and they add differing amounts of COD-Y, and as you add more COD-Y, you get obvious points where the COD-Y is binding to the DNA, and it's in two very noticeable locations. So it's protected in these two locations. It's interesting because there's also two catabolite repression elements that are close by. Next, what they do is they figure out, okay, well, if that's the case, does GTP make a difference, and isoleucine, valine make a difference, and the combination make a difference? And it seems that the branched amino acids do help bind in those two sites. And if you add GTP, the mix of the three do help.
How is this experiment different than the other one?
The last one?
No.
This one was
Before.
just COD-Y, adding COD-Y, and in this one they're adding COD-Y with the
But they
three
already
compounds.
know, right, that in
Right.
the presence of branch, okay, they are just dissecting the.
They're doing more experiments to prove their point. Here's another experiment to prove their point.
Seriously?
Yeah.
I got the point.
Now, this one's just a slight different way of doing the experiment. They add CCPA and they find a binding site.
Yeah.
Now they add CCPA and COD-Y, and this region is totally bound to.
What is this region?
Um,
Promoter region?
that would be between 1 and 2. The promoter of, yes.
Okay.
Now if you bind COD-Y, it's a little bit in a different location, and if there's no protection, this is what you get. So both proteins are binding to this DNA. And they bind at the same time. Next, what they do is they take a look at the ACK promoter, and they're adding more amounts of COD-Y. They're comparing it with, so this is actual in vivo, I say in vitro, but I think this is actual growth after time. And they're trying to figure out that if you add more COD-Y, there's more expression. Next, in vitro transcription in the presence of COD-Y and CCPA. So here's ACKA with no protein, and if you add some CCPA, 1.5 micromolar, you get 1.6 relative amount upregulated. Add cod Y, it's upregulated, and if you add both, it's upregulated even more. So adding the two proteins, you get more of the expression of the promoter. In this experiment, they enhanced the conditions by adding GTP and the branched amino acids. So they're kind of helping the results along too, by making sure that that should work. So by doing this, they're showing that the acetate kinase depends on both those proteins. And that's it for that paper. Next paper, Fujita. So there's the review paper showing the way this regulates. So here's the catabolite repression with CCPA. The protein binds to either two proteins, like I was saying earlier, HPR, or another protein, CARH, and when those two proteins are together, it forms a dimer on the catabolite repression element, so you get the CRE element. And this is just CRES, meaning it's like an alignment of a whole bunch of them. And the histidine protein is involved in the glycolytic cycle, taking sugar from larger forms to pyruvate. Just a side note, histidine protein has a histidine in it, which is phosphorylated at the cell membrane, but when it's involved with CCPA, it's phosphorylated at a serine. So it's a different location on the protein that's being phosphorylated. So at a different time, it's phosphorylated differently. Next, this is the TCA cycle showing the location of CCPA and how it's regulating the TCA cycle, and also involves COD-Y. I just thought I'd show you what that looks like. In red, it's activating all of these systems. In blue, all of these proteins are being repressed by CCPA. Next, here's a cellular level of the regulation. And that's pretty much what the paper shows. And it talks about all of these systems. Very complicated, but very detailed work.
Yeah.
That's what you'd expect from a big review. Next.
It could be a common mechanism in all bacteria, no?
Any bacteria that has a proper TCA cycle, right? Mutans doesn't.
TCA cycle?
It's not a full TCA cycle. It's
Okay.
Here's a paper by a group from Rochester. Robert Quivy talks about Streptococcus mutans and this protein, this aminotransferase ILVE. So Brenda Lees Santiago wrote this paper on this aminotransferase and how it's really regulated by COP-Y and CCPA.
So in cellulose, it was amino kinase,
Acetate kinase.
acetate kinase, and it is here. It's aminotransferase.
There's an aminotransferase involved with these two.
Okay. So they're totally different enzymes, right?
But
mechanisms of action would be
these two
different
proteins interact with large cycles, so they might have similar locations, similar proteins.
Wow.
There's a picture at the end of this paper I want to show you,
Sorry.
comparing Bacillus subtilis and S. mutans. So it'll kind of come up again at the end, okay? So here, for branched chain aminotransferase, ILVE, I showed you protein for CCPA.
Oh my god. I know.
So
I'm
disrespectful.
Sorry. I
It's
was
fine.
listening. I heard you.
Yeah.
Question about the two enzymes being different. Doesn't mute people out. Mutes like background noise, which there is none.
It's the wind. We can hear the wind, right?
Yeah.
Okay, so what happens with transamination with ILVE? It can make energy using up the carbon and regulate NADH ratios, as well as recycle glutamate from alpha-ketoglutarate. It also produces branched chain fatty acids for cell membranes. So that's what ILVE is responsible for. Now, in this paper by Santiago, what she did was or I think it's a she looked at ILVE. In the region prior to ILVE is this sequence, and what she's done is she's made a box around the promoter area. Now, what she's also done is done an experiment where she's only looked at a part of the promoter, and that's kind of highlighted by the two brackets. And the reason that she did a truncated promoter was she wanted to look at what happens if you don't include the COD-Y box in an experiment. In letter B, you have the web logo for a COD-Y motif, and letter C, you have the CCPA consensus motif. So this would be the Cree element on the right and the COD-Y element on the left. Now, does COD-Y interact with the ILVE promoter? This is the question asked in this paper, or this figure. So what they do is they take the ILVE promoter and try and bind it with the protein COD-Y. In the first line you see there's no DNA K, but there is COD-Y. Oh, sorry, DNA K. This is their negative control. There should not be binding.
Okay.
No DNA K, but there is COD-Y, and there's no complex. This would be the control. I'm trying to remember. Mind if I take a look at the paper? Let's take a look. I'm trying to remember why lane 1 is different. Promoter region used for binding assays were the ATPase promoter. So this is the ATA promoter. Oh, and ILVE is added in the next 3 lanes. So there's no ILVE in lane 1, just COD-Y.
Yeah.
Now, as soon as you add ILVE, then you get a complex being formed.
You're not adding COD-Y then. So it's not a complex, it's just the
Protein.
ILV promoter.
So
Yes.
no DNA K, no COD-Y. This is just the promoter. Okay.
Yeah.
So this is just the promoter with DNA K.
Yeah.
And if you add COD-Y, it's bound up there.
Yeah.
Okay. And this is done radio labeling the DNA.
But did it, and so when they are using this, is it different from, so if you go to previous slide, so you said that the authors just took this part, right?
In box.
In boxed area.
Yeah.
And they removed the COD-Y box
In another experiment.
in another experiment, not
Yeah,
this
not this one.
experiment. Yeah.
This one's good.
I was thinking if they are removing COD-Y box, then
Not
now
yet.
COD-Y is okay.
Okay. Next, what we have is, okay, so here's the ATPase.
Yeah.
Here's the protein, COD-Y, and branched amino acids, but no ILVE promoter. And then once you add the ILVE promoter with no what are they called? No other
I got it.
ingredients. you get no complex being formed. But if you add DNA-K and ILVE being the branched amino acids, there's still no complex being formed.
But there's no cod Y there.
Now, if you add a little bit of cod Y, there is a little bit of a complex, but it needs the branched amino acids. And as you can see, once you add the branched amino acids, you get a nice complex formed. Next, in 7, if you add isoleucine, you get a really good complex. But leucine and valine don't give as good a complex. So it's even dependent upon which amino acid that you add to the whole mix. Next, so cod Y binding is not enhanced by GTP. This is a question that they come up with. Does GTP help it?
They're just repeating it in Streptococcus mutans now.
Right. So
And
they've done
with
it in
this
Bacillus, and this is in
Yeah.
mutans.
Why not? 2 for 1 deal.
So you get the complex again, isoleucine, cod Y, really nice form.
But
Next.
they are using a different enzyme here. They are using aminotransferase, but the Bacillus people, they used
Acetate kinase.
Yeah.
Yeah.
I think both of them are doing the same function, or maybe they are doing something in that pathway of
Right.
producing branching amino acids.
Right. Next, we have the cod Y box is required. So this is the experiment where they remove the box.
Yeah. And now they couldn't see the complex.
Right.
Brilliant. Geniuses.
You remove the binding site, you don't get a band. There's no cod Y
Of
band.
course.
Good, yeah.
Yeah.
It should be that way. All right.
That's true.
Now, they take a look at what's going on in 2 different media. So they do it in bovine heart infusion as well as FMC, which is the minimal media. B is minimal media. If you do this experiment with minimal media, you get enhanced expression. So it's induced by starvation.
Of course. Right?
Yeah, we would hope that,
Yeah.
because cod Y is important as a stringent response protein,
Yeah.
because it senses the amount of amino acids. If there's not enough amino acids, it's going to be upregulating its systems. Next, ILVE mutant
If you
is
go
going.
back,
Go ahead.
they're
This one?
also using different pHs.
Right. So at pH 7.
Yeah. How did you miss that?
Sorry. Well, at pH 7, expression is lower than it is at pH 5, and you can tell more about what's going on by using pH 5. For example, in the beef heart infusion, at pH 5, if you just add leucine, you get repression. If you add isoleucine, less repression. Or it's
Less
the
induction, actually.
less induction rate.
Yeah.
Thank you. But if you're adding starving conditions at pH 5, look at it's gone way up. Now, we do the same. We do another experiment at beef heart infusion and FMC, and the ILVE mutant is lowering the expression of the promoter. So if you have ILVE as your background strain and you're adding in part of the promoter, at pH 5 it's lower.
So it's auto-regulation?
Yeah. They're doing this type of experiment with a cat activity assay. The promoter is fused to cat.
But at pH 7 there is no difference.
Right.
It's only under acid stress that this is doing the auto-regulation thing.
Right. Next, they do a cat activity to show ILVE is regulated by the 2 proteins. Now in A, they do both a deletion of cod Y and CCPA. Notice cod Y, it's deleted. When cod Y is deleted, the expression of ILVE goes up. And when you delete CCPA, it goes down.
So there's more of CCPA function in its regulation than
At pH 5, yeah.
both pages.
At actually both pHs, you're right. Next, what they do is they take a deletion of COD-Y and compare that with an overexpression mutant, a complemented mutant of COD-Y. And the complemented mutant returns the expression back to wild type at both pHs, 7 and 5. And if you do this experiment in FMC, you get very similar results as what you saw at in A. Now here's their little model. So they're saying if you have glycolysis making pyruvate, pyruvates making alpha-ketoglutarate, and ILVE makes the branched amino acids. CCPA is activating ILVE, and the branched amino acids activates COD-Y, so you get acid adaptation. Virulence. Now, also, you get pyruvate forming lactic acid, and lactic acid also regulates ILVE. So the acidic environment is important. This is very similar to work already kind of seen in other bacteria. This is a Nature Reviews picture. So glucose regulates CCPA, regulates ILVB. Branched chain amino acids regulate COD-Y. GTPA regulates COD-Y, but not in the case of mutants, I guess, because that's what we showed.
Even in the case of Bacillus subtilis, they showed that GTP was not doing anything.
Not very much,
Yeah.
but it does something, but not much. Yeah. And the GTP forms pentaphosphate, or guanosine pentaphosphate, right? For making tRNAs. So this region is involved with the virulence and acid adaptation of mutants.
What concentration of branch and amino acids were they using?
I think it's 10 millimolar.
Okay.
Here's their model. So if you have a COD box binding to COD-Y, this is when COD-Y is active, it lowers the level of ILVE expression. Then if CCPA binds to a Cree element, it can remove the COD-Y from binding, or maybe if you get the lowered pH, it relieves the repression. So you get more of the ILVE transcribed. And branched amino acids, and when it's under starvation conditions, that inactivates the COD-Y and you get more ILVE forming the amino acids. Next paper. Prius talks about COD-Y activating a small RNA in Bacillus subtilis. Alright, so there's this small non-coding RNA called BSRF. It was found in a study where they did the Bacillus subtilis RNA and went through a whole bunch of them, A, B, C, D, E, F, and this is the sixth one on the list. Apparently, it's about 80 nucleotides long, and it is found in subtilis as well as another bacillus called amyloliquefaciens. They've found the -10, -35 site for BSRF, as well as exactly determine which nucleotide is responsible for its transcription, as you can see in the footprint assay right there. Alright, so what they're next doing is looking at the expression profile of the small non-coding RNA, and they're looking in complex media as well as minimal media. And as you can see in complex media, it goes up to a maximum and then decreases.
What OD is this? What wavelength? 600?
I can't read.
They are reaching to a OD of 8?
Let's see. Can you read it here? This is, no, up here. 560.
Still 560 is
Oh, yeah.
600, right?
Look at that. OD is 8.0.
And it's starting from 8.2.
Yeah,
Idiots.
that's overnights.
Oh!
The overnights is starting at 8, and they
Yeah,
dilute it, and then it grows,
See?
and stationary
Your OD
phase.
reaches till what? 2? And you were so proud of that. 8.
Well, bacillus is different. E. coli is different too.
Even E. coli doesn't reach 8.
It goes up to 4, I think, or 5.
I've seen 2.
Yeah. I don't think I've ever seen it higher than 1.5 or 2.
Yeah.
But these guys have it going way up there, don't they? Isn't that crazy? Okay, they are looking at the expression in complex media and in minimal media, also in sporulation media. So when you grow it in sporulation media, when it reaches the highest OD, it's actually the lowest expression for this small non-coding RNA. Then what they did was they used rifampicin to try and figure out its half-life, and they did the half-life in both rich and poor media, and figured in poor media it's about 23 to 28 minutes for the half-life of the RNA, and in rich media it's about 50 minutes. It also forms a nice complex with this high-frequency protein via HFQ. Next thing they try and do is figure out its secondary structure using different RNases, and these 3 different RNAs help determine the kind of structure you can get, what kind of loops you can get, and they found 3 different structures by doing this experiment, and that comes up with this model. Now, if you take a look at this and add COD-Y to BSRF, you find a different amount of expression. Here's wild type on the left, and then in the deletion of COD-Y, the expression is reduced. If you increase the OD, wild type is expressed at this level, then it's reduced again. So that BSFR depends on COD-Y for its expression, and this also includes with glucose or without glucose. And they have this binding site for the COD-Y region. So they also do the experiment including branched chain amino acids as
Why
well.
are they using 23S ribosomal RNA but not
5S or 16S.
Is it new? Or
It's just a different. That's right. It doesn't matter which ribosomal unit you're using to do a northern, as long as you can resolve the band. But yeah, that's huge. I wouldn't use it. I would use something much smaller, especially if their product is only 80. Okay, they do a bunch of transcription assays, and what they're doing is they're cutting off the COD-Y site and showing that the COD-Y site has a beta-galactosidase activity of about 500 with glucose, but without glucose it's about half. Now if you cut off the COD-Y site, you get almost the same amount of expression. Now if you take the CRE site, or the CCPA site, and cut that off, initially it's the same, but the closer and closer you get to the -35 region of this small non-coding RNA, you're reducing its expression. So it's depending, the expression is depending on the length of that insert. So in this case, there's arguing that it's activated by glucose, because you can see these numbers on the left are higher than the numbers on the right. That's what they found. Also, what they found is branched chain amino acids, GTP and COD-Y, also enhance the expression of BSRF, sorry. So that's that paper, I think. Yep, that's it. Any more questions? No?
What does this small RNA does? Does it has any function? Or
Well, so far they figured out that it interacts with COD-Y. All that for interacting with COD-Y.
what journal is this?
Journal of Bacteriology, 2009.
Who is the group?
In Germany. It's a German group. Heike
Much
Priest.
finding they have. That's so interesting. Not for any of its functions.
Thanks for listening.